Have you ever wondered if tiny pieces of ancient DNA are hiding inside all of us? Scientists have been digging into the genomes of people living today and finding some surprising connections to humans that lived tens of thousands of years ago. How did these fragments get here? What can they tell us about our past? But what if the DNA scientists uncovered doesn't match Neanderthals, Denisovans, or any known human ancestor we've ever discovered? So who was it? What scientists actually found? Have you heard that parts of very old DNA are still inside people today? Arun Dervisula and Sriram Sankararaman at the University of California, Los Angeles, studied genomes from West African groups, including Yoruba and Mende. Their work showed that some West African genomes carry DNA from an archaic human group for which no fossil genome exists. Researchers describe that source as a ghost archaic population because no matching ancient bone has been sequenced. Their analysis estimated that some West African genomes include roughly 2 to 19% of this deep ancestry. That range may seem large, yet it points to a substantial legacy of mixing between different human branches long ago. The story moves to high places. Elisa Huerta Sanchez and collaborators compared DNA from people who live on the Tibetan Plateau to ancient sequences. They linked a particular version of the EPAS1 gene in Tibetan people to DNA related to Denisovans, an archaic human group first identified from remains in Denisova Cave in Siberia. The EPAS1 variant affects how the body handles low oxygen. People who carry it avoid making too many red blood cells at high altitude. That specific match shows a clear case where DNA from an archaic source contributed a trait that changed how a modern population copes with a harsh environment. Ancient viral traces appear in a different way. Renata Ferreira and Marcelo Briones reviewed old Neanderthal sequencing data and reported short DNA fragments that match viral families such as adenovirus, herpes virus, and papillomavirus. Some fragments came from Neanderthal bone recovered at Chagaskaya Cave in Russia. The fragments are short and chemically damaged, but they display patterns that match what happens to DNA after long burial. These viral bits do not mean modern viruses and ancient ones are identical. They indicate that ancient hominins carried viruses from the same broad families that still circulate today. Recovering such fragments gives a glimpse of infections faced by our relatives tens of thousands of years ago. A third route for ancient viral material is built into our own genome. Human endogenous retroviruses are viral sequences that were inserted into the genomes of distant ancestors millions of years ago. Some of these insertions remain in modern human DNA. Rodrigo Duarte and Timothy Powell at King's College London measured expression of certain endogenous retroviral sequences in human brain tissue. Their work reported associations between the expression of specific viral elements and conditions such as schizophrenia and other brain differences. Those associations are not conclusive proof of cause, yet they show that ancient viral DNA can be active in the body and may influence gene regulation. Major reference genomes anchor many of these comparisons. Researchers at the Max Planck Institute for Evolutionary Anthropology under Svante Pääbo produced the first high-coverage Neanderthal genome. Later, the same institute published Denisovan sequences from remains in Denisova Cave. These ancient reference genomes allow direct comparisons. They made it possible to identify Denisovan-like matches in modern samples and to trace many non-African genomes to Neanderthal ancestry. The types of ancient DNA that appear in living people fall into three broad categories. The first is direct ancestry from other human groups. For example, many non-African genomes carry Neanderthal segments. Some people in Oceania and parts of Asia carry Denisovan segments. West African genomes show material that traces to the ghost archaic source described earlier. The second category is short viral fragments recovered from old bones and sequencing archives. These fragments record which virus families infected ancient hominins. The third category is endogenous viral elements that integrated into ancestors' genomes and continue to exist in modern human DNA. Every item mentioned involves named people and places. The ghost archaic signal involves Arun Dervisula and Sriram Sankararaman at the University of California, Los Angeles, and modern samples from groups such as Yoruba and Mende. The EPAS1 link was reported by Elisa Huerta Sanchez and colleagues and tied to Denisovan material from Denisova Cave in Siberia. Viral fragments were reported by Renata Ferreira and Marcelo Briones from Neanderthal sequence data linked to Chagaskaya Cave in Russia. Endogenous retrovirus expression work was reported by Rodrigo Duarte and Timothy Powell at King's College London. The Max Planck Institute for Evolutionary Anthropology and Svante Pääbo appear repeatedly because their ancient genomes provide the benchmarks used across studies. The strength of the evidence varies. The EPAS1 case shows a strong match to Denisovan-like DNA plus a plausible biological effect on oxygen use at high altitude. The ghost archaic signal in West African genomes rests on statistical approaches that detect deep ancestry inside modern genomes without a matching ancient bone. The viral fragments from Neanderthal data are short and damaged, which means more samples and more analysis will help confirm their exact identity. Endogenous retroviruses are well documented across mammal genomes, yet linking specific expressed elements to health conditions requires further work. These reports together show that modern genomes carry a record of past contacts. Archaic human groups contributed DNA that remains in many populations. Viruses left traces both as short fragments in ancient material and as long-term insertions that became part of the genome. In 2020, Arun Dervisula and Sriram Sankararaman published their analysis and estimated roughly 2 to 19% archaic contribution in some West African genomes. The Neanderthal Genome Draft was published in 2010 under Svante Pääbo. The EPAS1 link to Denisovan-like DNA appeared in 2014 through work led by Elisa Huerta Sanchez. More African samples will clarify the story in the coming days. But if our DNA still whispers secrets from ancient beings, what hidden method allowed scientists to uncover these buried genetic echoes? How they found it so? How did scientists actually pull ancient DNA out of people's bones or even modern DNA and figure out that it came from very old human groups or viruses? The process is more than just magic, as it involves careful lab work, computer analysis, and smart statistics. First, they start with samples. For ancient humans like Neanderthals or Denisovans, scientists use bones and teeth from archaeological digs. These often come from important sites such as Denisova Cave in Siberia or Chagaskaya Cave in Russia. For modern human genomes, researchers use blood or cheek swabs from living populations, for example, West African groups like the Yoruba or Mende, or people from the Tibetan Plateau. When they pick which bone to sample, they choose dense parts such as the petrous bone or the inner ear because they trap DNA better over time. Next, the lab work begins. Extracting ancient DNA is tricky. Over thousands of years, DNA breaks down and chemical damage happens, the strands get cut into short pieces, and base changes accumulate. One common chemical change is called deamination. That is where cytosine, a DNA letter, loses an amine group and changes to uracil, which the lab machines often read as thymine. This happens especially at the ends of the short fragments. Scientists know that pattern well, and they use it as a signature of true ancient DNA. To deal with that, they run special cleanup steps. They use enzymes like uracil DNA glycosylase and other repair enzymes to remove uracil and repair some of the damaged sites. This makes the DNA more stable and more reliable for sequencing. Without that, you could easily misread ancient chemical damage as a real mutation. Once DNA is extracted and cleaned, they build a sequencing library, basically preparing the fragments so that a sequencing machine can read them. Because the fragments are so short, sometimes just 40 to a few hundred base pairs long, scientists use methods that favor these short bits. Then they sequence everything. Often, they do shotgun sequencing, which reads all bits of DNA randomly, or targeted capture, where they use probes to fish out certain parts of DNA, such as specific human or viral genes. After sequencing, the computer work starts. The short readings from the machine are matched back to a reference genome or viral genomes to see where they belong. For ancient human DNA, they compare it to human reference genomes or to Neanderthal or Denisovan genomes when available. When reading a map, they look at certain patterns to check authenticity. For example, they check for the damage signature. Those cytosine to thymine changes are especially near the ends. They also check how long the fragments are, Ancient ones are often shorter than contaminant DNA from living people. But what about the strange ghost archaic DNA that Dervasula and Sankararaman reported, without any reference to fossil DNA? Here is the clever part. They used a statistical method called a hidden Markov model, HMM. This helps them to scan across the genome of modern people and mark out segments that look very different as if they came from a deeply diverged branch. The HMM basically treats each region of DNA as archaic or modern and compares how dense the unique variants are, especially those not seen in outgroup populations. That way, they can spot archaic ancestry even if they do not have DNA from that archaic group. Another method is S', which looks for archaic variants in modern human genomes. It pulls out segments that likely came from Neanderthals or Denisovans by comparing variation patterns. This helps confirm introgression events in people. While they map DNA fragments and run these statistical scans, scientists also run contamination checks. Ancient bone samples might be contaminated by DNA from microbes or even from people handling the bones. To guard against this, labs use special cleanrooms, full-body protective gear, and strict protocols. They run negative controls, samples known to be blank, alongside the real samples to check for rogue modern DNA. They also estimate contamination using mitochondrial DNA, the small circular DNA in cells, because it is abundant. If the mitochondrial genome in the bone sample looks more like present-day humans than ancient humans, that signals contamination. They also compare damage patterns. Modern contaminant DNA tends to have long, undamaged fragments, while ancient DNA is shorter and damaged in ways that match what we expect after thousands of years. When researchers search for viral sequences, they take a similar route. Suppose they look into old Neanderthal sequencing data. They compare the short readings against a database of viral genomes such as adenovirus, herpes virus, and papillomavirus. When they find a match, they check for mapping quality. They ensure the fragment truly aligns well to that viral genome and that it does not align just as well to something else. Then they check for the ancient damage patterns on those viral readings. Is the fragment short? Does it show cytosine deamination? And is its length consistent with being very old? If it checks out, that suggests the virus was present in the ancient hominin. Because viral DNA in bone is usually rare, scientists repeat the analysis in multiple bone samples or sequencing runs. If they see viral fragments in more than one sample, that strengthens the argument that these were real infections long ago, not just lab contamination. For the human endogenous retroviruses, HERVs that live inside our own genome, they work a bit differently. They look at modern human tissue, like brain tissue, and run RNA sequencing to see which viral-like sequences are active. If a HERV sequence is being transcribed, it means the viral fragment inside our genome is not just dead junk, it might be doing something. Thanks for watching. If you enjoyed this video, hit like, leave a comment, and subscribe for more stories.